Hello students, how you all are? I hope you all are doing very good. Myself Archana, and today the chapter we are going to start is Sexual Reproduction in Flowering Plants. So let's continue with the chapter. When I'm talking that we are going to deal with the flowering plants, that means we are going to deal with the angiosperms, and that means we are going to deal with the flower, the reproductive unit in the angiosperms. So, when I talk about a flower, which is only the characteristic of angiosperms, it is not found anywhere else in the whole plant kingdom. So, when I talk about the flower, what are the different parts of a flower? It has a thalamus. It has a thalamus, which is called as receptacle also. And it has a stalk, which is called as the pedicel. Right, and over that it has sepals, it has petals, it has the stamen, and it has the carpel. Right, so these are the various parts of the flower we are going to deal with. Right, okay, just a second, there is a reflection on the board. Yes, beta. So now we are going to deal with the process of reproduction in the flowering plants. That means we are going to deal with these two parts, that is the stamen and the carpel. The petals and the sepals are the protective units of the flower. Sepals protect the flower, protect these inner walls, and the petals, they attract the insects, right? So we are going to deal with the stamen. So let's first deal with the stamen. The male reproductive unit. Stamen is the male reproductive unit in the plant, right? So let's draw a diagram of stamen, the typical diagram of a stamen. This is the filament and this is the anther. So what type of question comes from this portion regarding the structure of the stamen? So understand, beta, that whatever is present towards the thalamus, whatever part is present towards the thalamus, this part is the proximal part of the filament, and whatever is present away from the thalamus, this is the distal end of the filament. So remember this point, that this is the proximal end of filament. Okay, proximal end of filament, which is towards the thalamus, and this one is the distal end of the filament, which is away from the thalamus. So what question comes from this part, that the anther is attached to the filament from the proximal end or from the distal end. So what will be your answer? That the anther is attached. This is the line of NCERT, and the question also comes, right? Anther is attached to the filament by distal end, through its distal end. Okay, this is important line. And the filament is attached to the thalamus. The filament is attached to the thalamus via or through its proximal end. Okay, so this is the question which comes from this part, right? Now the filament has not the reproductive function. The reproductive function is present in the anther. Now what kind of question comes from the part of anther? Direct question comes. Direct question comes regarding the structure of anther. First of all, the structure of anther is tetragonal. What is the meaning of tetragonal? That it has four sides. It has four sides, tetragonal. Then second important thing, it is bilobed. It is bilobed. It has two lobes, one lobe, second lobe. Then it is dithecous, because in each lobe it has two theca. In each lobe it has two theca, and these theca will be representing microsporangium later on, right? So each lobe has two theca. Each lobe, we say it in context of each lobe. So each lobe has 2 theca, so diathecous, and then it is tetrasporangiate. It is tetrasporangiate. That means it has a total of 4 microsporangium within it. Okay. So if anyone asks you what is the nature of anther, you will say that in angiosperms, the nature of anther is tetragonal, bilobed, diathecous and tetrasporangiate. Okay, clear? So this is important. And it has a total of 4 microsporangium in it. Okay. Then later on, these microsporangium, when the anther matures, then these microsporangium are called as pollen sacs. An anther has 4 microsporangium, and when the anther gets matured, these microsporangiums, they are called as pollen sacs. This is important line and must be remembered, right? When the anther gets matured, the microsporangium are called as the pollen sacs. Okay? Okay. Now let's discuss. Now we understand what is the, and yes, one more thing, that a stamen can also be called as microsporophyll. The another name of stamen can also be called as microsporophyll. Okay? So, stamen has this typical structure, filament, anther. Filament is attached to the thalamus via the proximal end and to the anther via the distal end. Then, the structure of anther, you know, tetragonal, bilobed, diathecous, tetrasporangiate. And when the anther gets matured, the microsporangium are called as the pollen sac. Okay. Now, what is important in anther is development of anther. How the anther gets developed in case of angiosperms. So, development of anther. The development of anther occurs through special cells called as the archisporial cells. These are the special cells which ultimately give rise to the anther. Okay, so these cells, they divide like this. They divide periclinally, that means horizontally, and give rise to 2 layers. The outer layer is called as the parietal layer and the inner layer is called as the sporogenous layer. That means these special cells, the archisporial cells, they ultimately give rise to 2 layers. The outer layer is the parietal layer, and this outer layer ultimately give rise to this parietal layer ultimately give rise to the anther wall. This layer after many divisions forms the anther wall, and this anther wall contains 4 layers, that is epidermis, which we are going to deal in detail. Epidermis, then comes the endothelium, middle layers and the tapetum. Middle layers and the tapetum. So these 4 layers ultimately forms the wall of the anther and protects the anther, and several other functions we will be going to discuss. Sporogenous layer ultimately give rise to the sporogenous tissue, and this sporogenous tissue ultimately forms the microsporangium, and within these microsporangium there will be development of microspores, and then those microspores will give rise to pollen grains. Okay, so this is how development of anther occurs, right? These special cells give rise to 2 layers. The parietal layer gives rise to the wall of the anther, and the sporogenous layer ultimately give rise to the pollen grains. Now let's discuss the structure of the anther. Right, so I've just told you that, yes, so this is a NCERT diagram, very beautiful diagram. Here you can see the filament and this is the anther, right? So ultimately, pollen grains will be formed inside the microsporangium, and then the anther will dehisce. It will dehisce and the pollen grains will come out of it, right? See here, pollen sac is written. Instead of microsporangium, pollen sac is written. Why? Because when the anther matures, the microsporangium are called as the pollen sac. Okay? Yes. Now, this is the typical diagram of the anther, which is important. So here you can see beta that there are 1, 2, 3, 4 sides. That is why tetragonal structure. It has 1, 2, 3, 4 microsporangium. That is why tetrasporangiate. Now, I have just told you that the anther has the anther wall, the wall which has 4 layers in it, right? So, if, see, this is the part here. We are focusing on the anther wall. So this is the anther wall, the wall of the anther. And when we are focusing on it, then we see this. Then the outermost layer is the epidermis. Inner to epidermis, there is endothelium. Inner to endothelium, there are middle layers. Inner to middle layers, there is the tapitum. And finally, inner to tapitum, there is the sporogenous tissue, which ultimately forms the microspore mother cells. So if someone says that arrange the layers of anther wall from outside to inside, what you will say? First, the outermost layer of the anther wall is epidermis, then endothelium, then middle layers, and then tapitum. Okay, and finally, inside microsporangium, microspore mother cells are developed from sporogenous tissue, and then from microspore mother cells, microspores, and then ultimately the pollen grains. And when the anther gets mature, the pollen grains, the anther, it gets ruptured and the pollen grains come out. Sorry. So let's first discuss the anther wall. Let's first discuss the anther wall. And when I talk about the anther wall, the outermost layer, the first layer is the epidermis. So what is important regarding epidermis is that it is a single layer structure. It is single layered, and epidermis is protective in nature. The outermost layer of the anther wall is single layered, which is protective in nature and is called as the epidermis. Okay, then below the epidermis comes the endothelium. Below epidermis comes the endothelium. Endothelium again is single layered, and what is important regarding endothelium is that there is some deposition of special material on the walls of endothelium, and this is the deposition of alpha cellulosic fibers. The cells of endothelium has alpha cellulosic fibers. And what is the nature of these alpha cellulosic fibers? They are hygroscopic in nature. What is the meaning of hygroscopic? That they are moisture absorbing, and they absorb a whole amount of water in them, and when the anther gets matured, they lose their water, and when they lose their water, then they get shrinked, and when they get shrinked, they pressurize the wall of the anther, and the anther gets ruptured. That means, what is the function of endothelium? What is the function of endothelium? It helps in the dehiscence. It helps in the dehiscence of anther. Right? Dehiscence means rupturing of anther. Because of this layer, the anther gets ruptured, it gets dehisced, and thus this is helpful in dispersal of pollen grains. When the anther will get ruptured, only then the pollen grains will come out. Right? So, endothelium, the main function is the dehiscence or the rupturing of anther. Then, let's discuss about the middle layers. The middle layers are approximately 3 to 5 layered. They are 3 to 5 layered and they are ephemeral. What is the meaning of ephemeral? That means short-lived. Short-lived. The middle layers, they are short-lived. That means when they get formed, after a short point of time, they get degenerated. And when they get degenerated, they help in the nourishment of developing pollen grains. So they are short-lived and help in providing nourishment to the developing pollen grains. And help in providing nourishment to the developing pollen grains. Okay, so this is important regarding the middle layers. Now, the most important, the last but not the least, that is tapetum. Now, tapetum, what is important about tapetum? First of all, beta, as you have read the plant kingdom, now you know what is a sporophyte and what is a gametophyte, right? Until and unless I will do meiosis in any cell, the cells will be the part of sporophyte. That means the stamen is a part of flower and flower is a part of sporophyte. That means every part of stamen is diploid. Every part of stamen is a part of sporophyte and it will be a diploid structure until and unless we will perform meiosis in it. So, obviously, epidermis is a diploid structure, endothelium is a diploid structure because all of them are part of sporophyte. Middle layers diploid, but tapetum is polyploid. It is polyploid, or sometimes it is also called as triploid, right? That means something which has ploidy level greater than diploid ploidy level, it is termed as polyploid, right? And moreover, the cells, all the cells of the epidermis, endothelium, middle layers, each cell contain only one nucleus. That means they are uninucleated. But the tapetum, the cells of tapetum, they are multinucleated. They are multinucleated, or in NCERT it is written as binucleated. That means the cells of the tapetum, they have more than one nucleus in them. So these 2 points are very important regarding tapetum, that the ploidy level is polyploid and it contains more than one nucleus in each cell. And what is the reason behind it? The reason is failure of cytokinesis. That means in a cell, when karyokinesis will occur, that means division of a nucleus will occur, and just after that division of cytoplasm will not occur, that means it will be going to be multinucleated. Okay, so these 2 points are very important regarding tapetum, beta, very important. Now, what is the function of tapetum? What is the main function of tapetum? Providing nourishment to the developing pollen grains. It is the major function, nourishment to pollen grains, right? This is the major function of the tapetum. But apart from that, there are several other functions of the tapetum. And what are those? What are the other functions of tapetum? Let's see. This tapetum is also responsible for formation of pollen kit. The second function is formation of pollen kit. Now, what is this pollen kit? See, beta, you know basic structure of a pollen grain. It has intine, it has exine, right? A pollen grain has intine and exine, right? Every pollen grain. But those flowers which are pollinated by insects, they have an additional layer around the exine. They have an additional sticky yellow colored layer around the exine, and this is called as pollen kit. This is a sticky yellow colored covering around the exine, which is present only in entomophilous flowers. This kind of covering around the exine is present only in entomophilous flowers. That means the flowers which are pollinated by the insects only. They have pollen kit. And the pollen kit, how it helps? When the insect sits on the flower, because the pollen grains are sticky, the pollen grains get stick to their legs, and when they move, the pollen grains move with them. So pollen kit is formed by tapetum. Then third function is, it also helpful in the formation of Ubish bodies. It is responsible for the formation of Ubish bodies. And these special Ubish bodies, they in turn forms a very special chemical that is called as sporopollenin. Sporopollenin. This sporopollenin is the hardest substance, hardest and most resistant substance of whole universe. Literally nothing, no enzyme, no acid, no alkali, no pathogen can harm this substance, sporopollenin. So this is very resistant and very hard substance. And ultimately this sporopollenin forms exine, forms the outermost covering of the pollen grain, that is exine. So this is understood that ultimately the exine of the pollen grain is formed by tapetum. How? Because tapetum forms Ubish body forms sporopollenin, and sporopollenin form the exine of the pollen grain. Okay. Then what are the rest functions of tapetum? It is helpful in the formation of some hormones like auxin, IAA, which will be helpful in the growth of pollen tube later on. Then it is also responsible for formation of some enzymes like cellulase, pectinase and calase. These are some enzymes which are formed by the tapetum. And finally, it is responsible for the formation of compatibility proteins. These compatibility proteins are helpful in pollen pistil interaction. That means pollen carries the compatibility proteins with it, and when it lands on the stigma, it convinces the stigma that yes, I am the member of your species and please allow me to form the pollen tube. So it convinces the stigma with the help of these compatibility proteins. Okay, so these are the major functions of tapetum. Nourishment to pollen grains. Then it forms the pollen kit in the entomophilous flowers, then formation of sporopollenin, then hormones, enzymes and compatibility proteins. So this is about the anther wall. Now let's move to the microsporangium. We know beta that microsporangium is formed by a special tissue called as the sporogenous tissue. Right. Now what will happen? This sporogenous tissue, the cells, some cells of this tissue, this sporogenous tissue forms the microsporangium, and some cells of this tissue, they gets differentiated, right? Some cells of, obviously this is 2n, some cells of this tissue gets differentiated, they gets matured, they gets different from the rest of the tissue, and they form specialized cells called as the microspore mother cells. So this sporogenous tissue, after differentiation, it gives rise to special cells called as the microspore mother cells. Okay, right? So I'm telling you what is happening at the center. I've just talked about the anther wall. Now I'm discussing what is happening at the center where microsporangium is present. In that microsporangium, sporogenous tissue is present, which has formed specialized cells called as the microspore mother cells. Now we are going to study a special process occurring inside the anther, and that is microsporogenesis. That means formation of microspores in the anther. Formation of microspores inside the anther. Now let's see how these microspores are formed inside the anther. So inside each microsporangium, there are 4 microsporangium. So inside each microsporangium, there are many microspore mother cells. And I am taking only one of them out. So I am taking one microspore mother cells. And we will see that how microspore will be formed. So I am taking here single microspore mother cell. There are many microspore mother cells, right? So what will happen here in the microspore mother cell? Meiosis will occur. Okay, meiosis will occur, and what will be formed after meiosis in the microspore mother cells? There will be formation of 4 microspores. 4 microspores, and obviously these microspores are haploid because they are the result of meiosis. Now these 4 microspores, the microspore mother cell has a special type of compound in its cell wall. The cell wall of microspore mother cell have a special compound in it called as callose. Callose is a type of carbohydrate. So what will happen? One microspore mother cell, when it will undergo meiosis, it will form 4 microspores, right? And these 4 microspores, they remain attached to each other, right? These 4 microspores, they remain attached to each other. One will be behind it, right? So these are the 4 microspore mother cells which are remain attached. This is a tetrad. Tetrad means 4 microspores are attached to each other. 4 microspores, they remain attached to each other by, and what is the thing which is responsible that these 4 microspores are attached to each other is callose. Callose was present in the wall of microspore mother cell, and when it gives rise to 4 microspores, these 4 microspores remain attached to each other and they form a tetrad. And why they are attached to each other? Because of this compound callose. Okay, so here what kind of shape you can see here? This is the isobilateral shape. This is the isobilateral shape which is seen in case of monocots, right? But the shape of the tetrad can be tetrahedral also, and this is seen in case of dicots. Okay, so what is happening at the center? The sporogenous tissue, it gave rise to microspore mother cell, and each microspore mother cell undergo meiosis to form 4 microspores, and those 4 microspores are haploid structures which remain attached to each other and they form a tetrad. Now from these microspores we have to form the gametophyte. If you have attended the lecture of plant kingdom, I have told you that what is the function of spores. The spores, they give rise to gametophyte. Now these microspores, they will give rise to male gametophyte. And who is the male gametophyte? The pollen grain. So now let's discuss development of male gametophyte. Development of male gametophyte. And what is the another name of male gametophyte? Pollen grain. Microspore will give rise to pollen grain, which is a male gametophyte. So what are the changes that will occur while the microspore? Now first of all, when the 4 microspores, they are attached to each other, I have just told you, the 4 microspores, they are attached to each other because of the callose. Now, what will happen that there will be an enzyme which will come, and that enzyme is calase. And you know who have formed the enzyme calase? The layer tapetum. The calase enzyme will dissolve the callose, and when this calase enzyme will dissolve the callose, these microspores will be separated. These microspores will be separated from each other like this. They will be separated, and each microspore will develop into a pollen grain. Each microspore will develop into a pollen grain. Right. That means one microspore mother cell ultimately give rise to 4 pollen grains. Now, let's see that what are the changes that a microspore mother cell show when it ultimately give rise to pollen grain. So, in this diagram, beta, this is a microspore, a single microspore. Right? This is a single microspore. Okay, this is the diagram of NCERT, which is important. So, right now we are discussing development of male gametophyte or formation of pollen grain. First of all the 4 microspores, they got separated from each other, and then ultimately each microspore will develop into a pollen grain. So, how each microspore forms the pollen grain? Let's check it out. So, this is a microspore, right? A haploid structure. Now, the first change that occurs in the microspore is the development of vacuole. So, the first change occurs is vacuolation. The first change is vacuolation. That means a vacuole is developed in the microspore. So, nucleus is pushed towards periphery, towards side. Now, what will happen? Spindle fibers will be formed. That means asymmetric mitosis will be seen here. Asymmetric or unequal mitosis will be seen because of the formation of asymmetric spindle. The spindle formed towards the upper side, they are larger, and the spindle towards the lower side, they are smaller. Because of the formation of asymmetric spindle fibers, the mitosis over here is unequal, and because of that, one single microspore gets divided into 2 cells. And these 2 cells are unequal. They are not equal. Why? Because the unequal mitosis has just occurred. So ultimately, the microspore first undergo vacuolation, then unequal mitosis occurs in microspore, and then it forms a 2-celled structure. Then it forms a 2-celled structure, and this 2-celled structure is called as a pollen grain. A pollen grain or the male gametophyte. So this is how a single microspore gets changed into a male gametophyte. So what are you seeing over here? It's a 2-celled structure which is called as a pollen grain. The above cell is called as the vegetative cell, and the smaller cell is called as the generative cell. Right? Now, when the pollen grains gets formed inside the anther then is ready for rupturing, for dehiscence. And which layer was responsible for dehiscence? Endothelium. So anther will get ruptured and the pollen grains will come out. This is called as shedding of pollen grain. That means when they come out of anther. So in 60% of angiosperms, shedding of pollen grain pollen grain occurs at two-celled stage. When the pollen grain, it has two cells within it, then the anther will get ruptured and the pollen grains will come out. So in 60% of angiosperms, it is like that. But in 40% of angiosperms, shedding of pollen grain pollen grains occur at three-celled stage. That means when the pollen grain, it will be a three-celled structure. After that, the anther will get ruptured and the pollen grains will come out. So this is an important point. In 60% of angiosperms, it occurs at two-cell stage, and in 40%, it occurs at three-cell stage. Now, what is the meaning of three-cell stage? What happens that this generative cell again undergoes mitosis again. So this generative cell, when it will undergo mitosis, it will form two cells, and those two cells are called as male gametes. So finally, how many cells will be there in the pollen grain? Three. One vegetative cell, and the generative cell has given rise to two male gametes. So ultimately, the pollen grain is going to be a three-celled structure in all the angiospermic plants. But what happens that in 60% of angiosperms, when the pollen grain lands on the stigma and forms the pollen tube, after that, the generative cell divides to give two male gametes. But in 40% of the angiosperms, this happens inside the anther only. You got the point? Okay. In 60% of the angiosperms, the three-cell stage occurs when it lands on the stigma. But in 40% angiosperms, the generative cell divides to give two male gametes inside the anther only. Okay? Now, some important points regarding the generative cell and the vegetative cell. Okay, so see here, this is very important. Now let me first draw a diagram of a typical pollen grain. If I draw the diagram of a typical pollen grain of angiosperms, this is this. The outermost layer is called as the exine. And you know, exine is composed of which substance? Sporopollenin. And I have just told you that sporopollenin is formed by tapitum. Right? And this is the most hard substance of the whole universe. Then this is the point where sporopollenin is absent, right? Because it has to give away the way for pollen grain to come out, pollen tube to come out, right? So there is a space in the exine from which the pollen tube will come out, and the point where sporopollenin is absent, the point where sporopollenin is absent in exine is called as germ pore, right? Is called as germ pore. So this germ pore is the point in the exine where sporopollenin is absent, and from this pore, pollen tube will come out. From this pore, pollen tube will come out. Will come. Okay. Now, the inner layer is the intine. The inner layer is the intine. And this intine is pectocellulosic, because it is composed of pectin and cellulose. So this is pectocellulosic in nature, and it gives rise to pollen tube. Extension of intine is pollen tube. What you have to do, if you take this intine and stretch it, then pollen tube will be formed. Then pollen tube is nothing but extension of intine. That is why the nature of pollen tube is also pectocellulosic. So extension of intine is pollen tube. Extension of intine is pollen tube. The smaller cell is the germ cell and the larger cell is the vegetative cell. So this larger cell is called as the vegetative or the tube cell. And what is the function of this vegetative cell or tube cell? It will form the pollen tube later on. And this smaller cell is called as the germ or the generative cell. And what is the function of generative cell? To give rise to two male gametes after mitosis. So this is the typical structure of pollen grain. Let's see what NCERT has to say about this. When the pollen grain is mature, see the lines. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. Okay, the vegetative cell is bigger. Okay, learn these words with me. The vegetative cell is bigger, has abundant food reserve, right? That means the food reserve in the pollen grain is due to the presence of vegetative cell. It has a large irregularly shaped nucleus. So what are the three properties of vegetative cell? According to NCERT, it is a larger cell. It has abundance of reserved food material and the nucleus of the vegetative cell is large and irregularly shaped. Now, the generative cell is small and floats into the cytoplasm of the vegetative cell. The generative cell is so small and it has very less amount of cytoplasm that this generative cell floats in the cytoplasm of the vegetative cell. Now, what is the shape of the generative cell? Generative cell is a spindle shaped. Spindle shape is not the shape of the nucleus of the generative cell, but it is the shape of the cell of the generative cell. Okay, and it has dense cytoplasm and a nucleus. Okay, this I have told you. So are you clear with it? Clear? Okay. So what we can say here that for two-celled stage of pollen grain, for two-celled stage of pollen grain, if I talk about the two-celled stage of pollen grain, then how many meiosis and how many mitosis will be required? See, if you have to form a two-celled pollen grain, then how many meiosis and how many mitosis you will have to do? Answer. So for two-celled stage of pollen grain, one meiosis and one mitosis is required. Remember, meiosis, why? Microspore mother cell will undergo meiosis to form microspore, and then in that microspore, mitosis will occur to form a two-celled pollen grain. Then for three-celled stage of pollen grain, that means pollen grain, that means the pollen grain, when it will contain two male gametes, then how many meiosis and mitosis will be required? One meiosis and two mitosis will be required. Why two mitosis? Because by first mitosis, generative cell and vegetative cell will be formed, and after second mitosis, the generative cell will divide to give two male gametes. That is why two mitosis. So remember this. For two-cell stage, one meiosis, one mitosis. For three-cell stage, one meiosis, two mitosis. Okay. So now we have formed the pollen grains, and when the pollen grains are formed inside the anther, this is the line of NCERT. When pollen grains are formed inside anther, then what are the changes that occur in the anther? When pollen grains formed inside anther, then the anther will dehydrate. That means it will lose its water. And when it will lose its water, there will be shrinkage of cells and the anther will rupture. The anther will dehydrate and there will be dehiscence of anther, rupturing of anther. And which layer will be helping it out? Endothelium, right? And then the pollen grain will be dispersed. Okay, so this is how. Now what we have understood up till now, what is stamen, what is anther, what is the structure of anther, how anther wall is formed, what are the 4 layers present in the anther wall, what are their functions, what is their nature, and then how microspores are formed inside microsporangium and how those microspores give rise to the male gametophyte, that is pollen grain. Now, there are certain things which are given in NCERT regarding the pollen grain, and they are the pollen allergy. Pollen allergy. The pollen grains of some plants are allergic. They cause bronchitis or asthma, right? And this phenomenon is called as pollen allergy. And direct question comes, the examples of those plants whose pollen grains can cause bronchitis or asthma, and the examples are Amaranthus, Chenopodium, Amaranthus, Chenopodium, and Parthenium. Parthenium is also called as the carrot grass. The carrot grass, okay? So important, Amaranthus, Chenopodium, and the Parthenium. Then comes the pollen products. Pollen products, as the pollens are rich in nutrients, I have just told you that the vegetative cell has enough amount of reserved food materials. So the pollen grains, they are very rich in nutrients, and nowadays it is fashionable. The pollen grains are available in the form of cough syrups, they are available in the form of tablets, and these pollen grains are taken by the athletes and the horses to increase their performance. So these are the pollen products. Then comes the pollen banks. What are the pollen banks? The pollen grains of certain species, they are preserved in the laboratories for various experiments, and this is called as a pollen bank. And what is done in the pollen banks? The cryo preservation. What is cryo preservation? The pollen grains are preserved at minus 196 degrees centigrade. Pollen grains are preserved at minus 196 degrees centigrade in liquid nitrogen. This question comes, very important. The pollen grains are preserved at minus 196 degrees centigrade at liquid nitrogen. This is cryo preservation. And then comes the important thing, that is pollen viability. Pollen viability. What is pollen viability? That when the pollen grains, they come out of the anther, then the time period up to which they are capable of forming pollen tube, that time period is called as the pollen viability. Okay, and the pollen viability is different in different plants. Like in cereals, it is given in NCERT that in cereals like wheat, rice, right, the members of the Graminea family, they have pollen viability of just 30 minutes, half an hour. That means when the pollen grains comes out of the anther, then within half an hour it has to reach the stigma. If it doesn't, then it is wasted. It will not be able to form pollen tube after that. Then the pollen viability can be of several months. That means after coming out of anther, the pollen grain is capable of forming the pollen tube even after several months. And the examples given in NCERT are 3 families: the Solanaceae family, that is the potato family; the Rosaceae family, that is the rose family; and the Leguminaceae family, that is the pea family or the soya bean family. These families, these 3 families has the pollen viability for several months. Okay, so this is regarding the male reproductive part. Now let's move on to the female reproductive part. and the female reproductive part is represented by the carpel and also called as the pistil, right? And the collection of carpel or pistil in the flower is called as the gynoecium. It is called as the gynoecium. Now, what sort of questions comes from this part? Okay. Now, according to NCERT, the gynoecium can be monocarpillary. Monocarpillary means when the gynoecium contains only one carpel, right? It has only one carpel in it. But it can be bicarpillary if the gynoecium contains two carpels, tricarpillary, tetracarpillary, up to and so on, right? So it can be up to and so on. More than two is counted in multicarpillary. More than two is counted in multicarpillary, that is tri, tetra, penta, right? So if there are two carpels, within the same flower, right? Two carpels. That means if there are two carpels in the gynoecium, then they can be fused or they can be free. If there are 3, 4, 5, that means if there is more than one carpel in the gynoecium, then they can be either in the fused condition or in the, so if this condition is there, because single, if it is single, then how it can be fused or free? It will be free. But if the gynoecium contains more than one carpel, then it can be either fused, the carpels present in the gynoecium, they can be fused or they can be free. If they are fused, this condition is called as the syncarpous condition, and if they are free, this condition is called as the apocarpous condition. And this makes the important question. This is the most common condition found in the flowering plants when the carpels in the gynoecium, they are present in the fused condition. But when they are present in the free condition, there are very few examples. One is the example of Michelia, lotus, rose, etc. These are some examples of the plants in which there is multicarpillary gynoecium, and the carpels in the gynoecium, they all are free from each other. And the most common condition is when the carpels are fused with each other. So this makes an important question from this part. Here is the diagram given in the NCERT. Right. So this is the last diagram. This one is the diagram of Michelia. And what is important about Michelia, that it has multicarpillary apocarpous gynoecium. Multicarpillary apocarpous. That means it has many carpels in a gynoecium and all are free from each other. All are free from each other. And in case of the rest two examples are of hibiscus. Hibiscus, also called as China rose, and papaver. Papaver somniferum. So in both papaver and hibiscus, in papaver and hibiscus, hibiscus is the China rose. In both of them, the gynoecium is multicarpillary and syncarpous. That means fused. And the most common condition is fused, right? So this makes the important question from this part. Okay. Now let's discuss the structure of a carpel or a pistil. If I draw the diagram of a carpel or a pistil, then you know this is a typical diagram. This part is a stigma on which the pollen grain comes and lands on. This is the style through which the pollen tube travels, and this is the ovary. Okay, so the ovary, the space, this is ovary is hollow from inside, and the space within the ovary is called as locule. Right, and this locule, if there is single space, then unilocular. If there is two spaces, bilocular, multilocular, trilocular, okay? And within this space, what are present? Within the space of the ovary, what important structures are present? Ovules. Ovules are present, which are also called as megasporangium. Inside the anther, microsporangium were present, but inside the ovary, megasporangium are present, which are also called as the ovules. Okay, and how ovules are present here? They are attached with the wall of the ovary with a special structure called as funicle. The ovules are attached with the wall of the ovary with a special structure, with a special stalk-like structure called as funicle. Okay, so an ovule is present inside the ovary, and an ovary can have single ovule. An ovary can have a single ovule. Inside an ovary, there can be single ovule or there can be many ovules. It depends from plants to plants. Okay, all those plants which have single seeded fruits, all the cereals, mango, they have single ovule within the ovary. And all the plants which are many seeded like papaya, watermelon, that means their ovary has many ovules. Okay, so this can be the example of cereals and mango. And here there are several examples, papaya, watermelon, etc. Right? Okay, now we know it. Now let's move on to ovule. Let's have a typical diagram of ovule, and this is the typical diagram of an ovule. An ovule in case of angiosperm is also called as integumented megasporangium. Integumented megasporangium. That means the megasporangium which has protective covering around itself is integumented ovule. So this integumented ovule has, and yes, what is the nature of a typical angiospermic ovule? It is anatropous in nature. What is the meaning of anatropous? That means the ovule body gets rotated by 180 degree. The ovule body gets rotated by 180 degree. The ovule body rotates by 180 degree over funicle. This is anatropous ovule. So a typical angiospermic plant has anatropous ovule, and these are the several parts of the ovule. Let's understand. This is the funicle. This is the funicle or the stalk of the ovule, with the help of which I've just told you, the stalk of the ovule, with the help of which the ovule gets attached to the wall of the ovary. So this is the stalk of the ovule called as the funicle. Now the point at which the ovule body is attached to the funicle is called as hilum. This point is called as hilum. The point of attachment. What is hilum? This question directly comes. Point of attachment of funicle with ovule is hilum. So hilum is point of attachment of funicle with ovule. That point of attachment is called as hilum. Okay. Now the ovule has two coverings, the outer integument and the inner integument. That is why it is called as a bitegmic structure. It has two integuments. Then inside the integument, it has a mass of parenchymatous cells, which is called as nucellus. And ultimately, when the ovule gets matured, embryo sac formed inside the ovule, right? The chalazal end, the base of the ovule, is called as chalazal end, and this is the opening which is called as the micropyle. So this is a typical structure of the ovule, right? Funicle is the stalk. Hilum is the point of attachment of funicle with ovule. It has two integuments. Inside the ovule, there is a mass of parenchymatous tissue called as nucellus, which ultimately give rise to embryo sac and micropyle and chalaza. Okay. Now, let's see what happens inside this ovule. Now, let's understand the process of megasporogenesis. Let's understand the process of megasporogenesis or formation of megaspore. Or formation of megaspore. So, we know that inside the ovule, nucellus is present, right? Nucellus is present, which is a diploid structure. So, let's take this. This is the ovule, right? This is the micropylar end. This is the chalazal end. Okay. Now, a mass of cells. This ovule is all filled up with a mass of cell called as the nucellus, which is a sporophytic tissue, a diploid tissue. Right. Now, what will happen? Listen carefully. Only one single cell of nucellus, only one single cell of nucellus towards the micropylar end gets differentiated and forms megaspore mother cell. This question directly comes. Megaspore mother cell. Now, what sort of question comes from here? A single cell of nucellus. A single cell of nucellus towards micropylar end. Towards micropylar end. Now, they confuse it. They will say that a single cell of nucellus towards the chalazal end forms megaspore mother cell. False. Megaspore mother cell is formed towards the micropylar end. So, a single cell of nucellus towards micropylar end gets differentiated to form the megaspore mother cell. And NCERT says that this megaspore mother cell has dense cytoplasm. This is large cell and it has a prominent nucleus. So, these are the properties of. See the difference. Inside the microsporangium in anther, many microspore mother cells were formed. But inside the megasporangium or ovule, only one megaspore mother cell is formed. And now what will happen? This megaspore mother cell will undergo meiosis, right, to form 4 megaspores. It forms 4 megaspores. But what happens that out of the 4 megaspores, the 3 towards the micropylar end gets degenerated. 3 of them degenerates and only this remains. Only one remains, and that is called as the functional megaspore. So out of the 4 megaspores, 3 gets degenerated and only one. And this is important. The functional megaspore is present towards chalaza. This is important. Megaspore mother cell was formed towards micropyle, but megaspore is formed. The functional megaspore is present towards chalaza. Now, from this single megaspore, we will form the female gametophyte. Remember, the microspore gave rise to the male gametophyte, that is pollen grain. Now, the single megaspore will give rise to the female gametophyte, that is the embryo sac. So, now let's understand formation of female gametophyte. formation of female gametophyte or embryo sac, which is a haploid structure. Now, what will happen? This functional megaspore will undergo many changes. So, this is a functional megaspore which was present towards the chalaza. It will undergo first pre-nuclear division. First pre-nuclear division. And what kind of pre-nuclear division is this? Mitotic division. What kind of free nuclear division is called as a free nuclear division? A division in which only the nucleus divides and not the cytoplasm. So this is a kind of mitosis in which only nucleus will divide, and just after the division of nucleus, the division of cytoplasm will not be there. So in the first mitosis, only the cytoplasm will divide. So it will be a binucleated structure. Two nucleus will be formed. Then second free nuclear division, second mitotic division, which is again will be free nuclear. That means only the nucleus will divide. So it will become tetranucleated. Then third mitotic division. Third mitotic division, again free nuclear. So it will become octanucleated, 1, 2, 3, 4, 5, 6, 7, 8. So ultimately this becomes an 8 nucleated structure. So in a functional megaspore, 3 free nuclear division occurs. 3 free nuclear mitotic division occurs to ultimately give rise to a 8 nucleated structure. And then what will happen in this 8 nucleated structure? Polarization. Polarization. What is the meaning of polarization? That out of the 8 nuclei, 1, 2, 3, 3 will go towards the, if I am taking it as the chalazal end. You can take any end as chalazal and any end as micropylar, but you have to remember what kind of cell are present towards micropylar end and what kind of cell are present towards the chalazal end, right? So if I am taking this as the micropylar end, 3 will go towards the micropylar end, and the 2 nuclei will remain at the center. So this is called as polarization. Then out of the 8, 3 will go towards the chalazal end, 3 nuclei will migrate towards the micropylar end, and 2 will remain at the center. Now what will happen after this? After this, cell wall development will happen. After this, cell wall development will happen. 3 nuclei are present this end, 3 nuclei at this end, and 2 at the center. So just see carefully what I'm going to write over here. Out of the 8 nuclei, this is the line of NCERT. Out of the 8 nuclei, 6 nuclei undergo cell wall formation. This is the line of the NCERT and important. Out of the 8 nuclei, 6 nuclei undergo cell wall formation. That around the upper 3, cell wall will be formed. Around the upper 3, cell wall will be formed. Around these lower 3, cell wall will be formed. So now there will not be called as nuclei, they will be called as cells because of the cell wall development. So what will happen with these two? The cell wall will not be formed around them? No, because the rest of the cell is theirs. Whatever the property has to be distributed, it was distributed amongst these 6, and the rest of the property is with these two nuclei. That means the rest of the cell is the cell of these 2 polar nuclei, and this is called as the central cell. So this is important line, that out of the 8, only 6 nuclei undergo cell wall formation, and what is the result? 3 cells toward chalaza. These are now cells, they are not nuclei now, they are cells, and 3 cells towards micropyle, and 2 at the center. This largest cell is called as the central cell. Is called as the central cell, having two polar nuclei. These are the three antipodals. These three cells are the three antipodal cells present toward chalaza. And this is a three-celled structure called as egg apparatus. Egg apparatus has one egg cell in it, one egg cell and two synergids. So the question asks, egg apparatus is how many celled structure? Egg apparatus is three-celled structure, contains one egg cell and two synergids, and it is present towards the micropylar end. And these are the two polar nuclei which is present in the largest cell of the embryo sac. Now the structure which are present over here, this structure is called as the embryo sac. The embryo sac, or also called as the female gametophyte. The embryo sac or the female gametophyte, right? So you can see, count the number of cells with me. 1, 2, 3, 4, 5, 6, and this is the largest cell, 7. So embryo sac is 7-celled and 8 nucleated structure. Why 8 nucleated? Because the central cell has 2 nuclei in it. And this kind of embryo sac is called as polygonum type of embryo sac. Why it is called as polygonum type of embryo sac? Because this kind of embryo sac was first studied in the plant Polygonum, right? And this kind of embryo sac is Monosporic Embryo Sac. This is very important. This kind of embryo sac is Monosporic Embryo Sac. What is the meaning of Monosporic Embryo Sac? That the embryo sac develops only from one megaspore. Develops only from one megaspore, because the rest 3 of the megaspores, they were degenerated, but the one single megaspore ultimately gave rise to the female gametophyte. So you have to remember these things about the embryo sac, right? It is a 7-celled 8 nucleated structure, it is polygonum type, and it is monosporic in nature. Now, what kind of question comes? For development of embryo sac, to form an embryo sac, how many meiosis and how many mitosis are required? So you have just seen that for development of embryo sac, 1 meiosis and 3 mitosis. Remember, 3 mitosis I have just shown you occurs. 1 meiosis, 3 mitosis. Okay. Now, what another sort of question comes, that what is the function of synergids in embryo sac? Synergids are also called as the helper cells. Why? Because they have a special structure in them. The synergids have a special structure present in them, that is called as the filiform apparatus. The filiform apparatus is present towards the micropylar end in the synergids. And what is the function of this filiform apparatus? To guide pollen tube into the embryo sac. To guide pollen tube into the embryo sac. That pollen tube has not to go anywhere but to come inside the embryo sac. Okay. Antipodals, they ultimately degenerates to provide the nourishment, right? So now we have formed the female gametophyte and the male gametophyte. The female gametophyte is called as the embryo sac, which contains the egg cell, and the male gametophyte is the pollen grain, which contains the male gametes. Now, what will happen? Fertilization. But before fertilization, what will happen? Pollination. So let's see. What is pollination? Okay, so let's see. So before fertilization, what we'll have to do? We'll have to disperse the pollen grains from anther to the stigma. So pollination is transfer of pollen grains from anther to stigma. Now this pollination can be further of three types. Pollination can be further of three types. It can be autogamy or the self-pollination. Autogamy or self-pollination, then getonogamy, and then comes the allogamy. Allogamy or xenogamy or cross-pollination. Okay. Now, what is autogamy? Autogamy means when a flower has stamen as well as the carpel, and the pollination occurs within the same flower. The pollination occurs within the same flower. So when I talk about autogamy, then there will be involvement of same plant and same flower. That means it's a plant and the pollen grain of the same flower will be landed on the stigma of the same flower. It will occur within the same flower. Within the same flower. Okay, within same flower. So it will be done by itself. Auto means by self. Nobody is required. Pollinating agent is not required. Even wind is not required. Wind is also a pollinating agent. That is also not required. Pollinating agent is not required. This kind of pollination occurs by itself. This kind of pollination occurs by itself because the pollen grain of the same flower lands on the carpel or the stigma of the same flower. Then what happens in getonogamy? That the plant is same, but the flowers are different. The plant is same, but the flowers are different. That means on the same plant, the pollen grain of this flower lands on the pollen grain of this flower. That means same plant, but different flower. When the pollination occurs between the different flowers of the same plant, then this is getonogamy. And obviously here, pollinating agent will be required. So here, pollinating agent is required. Okay. Cross-pollination means different plant, different flower. Different plant and different flower. This flower is present on different plant and this flower is present on different plant, right? So this is xenogamy, allogamy or cross-pollination, and obviously pollinating agent. Without pollinating agent, cross-pollination will not be occurring. So pollinating agent is required over here also. So these are the three types of pollination: autogamy, getonogamy and allogamy. Clear? Now what is important about getonogamy? A very important line, that getonogamy is functionally or you can say ecologically it is cross-pollination. Getonogamy is functionally or ecologically it is cross-pollination. Why functionally or ecologically it is cross-pollination? Because pollinating agent is required. It is not occurring by itself. But genetically the getonogamy is self-pollination. Why? Because same plant is involved. because same plant is involved and the genetics of the pollen grain will be similar to the genetics of the carpel. So genetically, geitonogamy is self-pollination, but ecologically it is cross-pollination. Okay. So now what kind of questions comes from this part? That what are the conditions that promote what? Self-pollination. Contrivances or conditions that promote self-pollination. All those conditions which promote self-pollination, they are called as the inbreeding device. Inbreeding devices. Why they are called as the inbreeding devices? Because the genetics, because in this context, if the plant will continue to self-pollinate, there will be no change in the genetics. So it will be inbreeding. So all those devices, all those conditions that promote self-pollination, they promote inbreeding depression, right? That is why they're called inbreeding devices. So what are the various inbreeding devices? First, bisexual flower. Bisexual flower, also called as the monoclinous flower. This is opposite. Remember, this is opposite. A monoclinous flower is a bisexual flower. Bisexuality of flower is very essential for self-pollination. If the flower is not bisexual, self-pollination will not occur, right? Second important thing is homogamy. What is homogamy? That both carpel and stamen mature at same time. Both carpel and stamen mature at same time. If they get mature at same time, then homogamy, then it will promote self-pollination. Then comes the cleistogamy. What is cleistogamy? Cleistogamy is a phenomenon where flower remains closed. That means the flower will never be opened up. And when the flower will always be remain closed, it will be never opened up, then it will obviously show self-pollination, right? So cleistogamy, what is the disadvantage of cleistogamy? The disadvantage of cleistogamy is that it may lead to inbreeding depression, because there will be no change in the genetics of the plant. There will be no variation. So it may lead to inbreeding depression. But what is the advantage of cleistogamy? That without the requirement of pollinating agent, there will always be formation of seed. There is assurance that seed will be formed even in the absence of pollinating agent, right? And the last one is the bud pollination. In some plants, when the flower is a bud, and it has not bloomed up, the reproductive parts, they get mature and they show self-pollination. This is called as bud pollination. This is often seen in pea plant and in rice plant. Okay, so these are the four conditions: bisexual flower, homogamy, cleistogamy, and bud pollination that promotes the self-pollination. Now let's see the conditions that promotes the cross-pollination. And all those conditions that promotes cross-pollination, they are called as the outbreeding devices. And what are the outbreeding devices? Let's see. First, unisexual flower. If a flower, that doesn't mean that a bisexual flower cannot show cross-pollination. I am citing a line, listen it carefully. A bisexual flower can show cross-pollination, geitonogamy, or self-pollination, but self-pollination will always be shown by bisexual flower. Are you getting my point? A bisexual flower can show self-pollination, cross-pollination, geitonogamy, but autogamy will always be seen in bisexual flower. And in unisexual flower, obviously geitonogamy and cross pollination can be seen. How? We will see it. So unisexual flower, cross pollination, then heterogamy. What is the meaning of heterogamy? That carpel and stamen matures at different time. They are not, they do not get mature at similar time. Carpel and stamen matures at different times. This is heterogamy. Then comes heterostyly. What is heterostyly? That means the position, there is very much difference between the length of the style and the length of the stamen, right? So it the pollen grain will not be able to, land on the stigma. So there will be difference between the length of stamen and the length of the style. So this is heterostyly. Then self-sterility. In some plants, the pollen grain of the stigma of the same flower do not recognizes its own pollen grain. So it will support cross-pollination. And then chasmogamy. What is chasmogamy? When the flower gets opened up. When the flower opens up or blooms, then it supports the cross-pollination. So these all are the outbreeding devices which supports the cross-pollination. Now in NCERT, there are three plants given. There are three plants given which have both the types of flowers in them, cleistogamous as well as chasmogamous, and the examples are viola, commelina, and oxalis. Right? In all these three plants, they have both the kind of flowers on them. They have chasmogamous which gets opened up as well as cleistogamous which do not get opened up. They have both the kinds of flowers in them. Okay, now let's see what kind of question comes from this portion. They will ask that in papaya and date palm, what is possible and what is not possible? In papaya and date palm, what is possible and what is not possible? I have told you this in the chapter reproduction, what I have, which I have just, taken the part, that there are two kinds of plants, monoecious, dioecious. A dioecious plant has only one type of unisexual flowers, but a monoecious plant can have bisexual flower, or it can have two types of unisexual flowers over the same plant. Right? I have told you this in the reproduction in plant, and I told you that it will be used up again in the chapter sexual reproduction. So papaya and date palm, they are dioecious plants. That means on a single plant, they have only male flower. On one plant, they will having all the flowers will be male flowers. And on the other flower, all the flowers on the other plant, all the flowers will be female flowers. So what is possible in them? What is possible in these kind of plants? Obviously, geitonogamy will not be possible, because geitonogamy happens between two flowers of the same plant, and when all the flowers are male flowers, then geitonogamy will not be possible. Obviously, autogamy will also not be possible, because for autogamy you must have bisexual flower. That means only one thing is possible, and that is xenogamy. This kind of questions comes very often. So in date palm and papaya, which are dioecious plants, only what is possible? Xenogamy. And then comes in maize and cucurbits, what is possible? So you'll say maize is a monoecious plant. A monoecious plant. They are monoecious plants which have unisexual flowers of both the types on them. Some flowers are male flowers and some flowers are female flowers. Yani ke they have unisexual flowers, but those unisexual flowers are present on the same plant. So apply your mind and tell me, will autogamy will occur or not? Autogamy. Obviously not, because autogamy happens only in bisexual flower, but they have unisexual flower. Geitonogamy will happen or not? Obviously it will happen, because they have male and female flowers both. So it will happen between the different flowers of the same plant. Cross pollination, yes, obviously there will be room for cross pollination also. Okay, so these are some famous questions which come from this part, from the pollination. Now let's talk about the pollinating agents. Pollinating agents. There are two kinds of pollinating agents, biotic or abiotic. Biotic are the living agents, right? And the most common type of pollinating agents is insect. Insect are the most common pollinating agents, right? And in the insects also, the bees are the most common. There are so many type of insects. On earth, there are 7 insects on every 10 organisms on the earth. So there are so many types of insects, but the bees are the most common pollinating agents. Insects, then there can be snail, then there can be lemurs, monkeys, elephant, humans. They all help in the snake, birds, humans. These all are the biotic pollinating agents, and the most common one are insects. Abiotic are wind and water. Wind and water. And amongst wind and water, wind is more common. Wind pollination is more common amongst wind and water, but the most common pollinating agent is insect. Okay. Now let's discuss each one of them one by one. First of all, let's discuss the wind pollination. Wind pollination is also called as anemophily. Do not get confused in it. Anemo word, not animal. It is wind pollination. Now, what are the properties of flower which shows wind pollination? This is important. Obviously, the flowers which show wind pollination, they will be odorless. There will be no aroma or smell in them. There will be nectarless, right? Nectarless, odorless, nectarless. Okay. Then they will be colorless also, because they need not to attract the insects. Then what is the important part? Their pollen grains are light. They are light so that they can easily move with the help of air. Light and non-sticky. Light and non-sticky. This is important. Then their stigma. Their stigma is well exposed towards the air so that it can easily receive the pollen grain. And the stigma should be feathery. It should not be sticky. It should be feathery. Feathery means it should have brush-like structures in it, so it can easily receive the pollen grains. So it should be well exposed and feathery. And what is more important is that the ovary of the flowers contains single ovule. This is very important point. Ovary contains single ovule always. All those plants which show anemophily, their ovary will always contain single ovule. This is a very important point. And the flowers are often packed into inflorescence. The flowers of the wind pollinated flowers, they are packed into inflorescence. Inflorescence is a group of flowers present on the plant. So these are the important points which the flowers which show wind pollination they have. All these points are important. Light and non-sticky, well exposed, feathery, contains single ovule. Okay, and one more thing, that wind pollination is a directionless and wasteful process. It's a directionless, there is no certain direction, and wasteful process, because there is no surety that the wind will take the pollen grains to the desired destination. So that is why all those plants which are anemophilous, they form a large amount of pollen grains. Okay, and their ovary has only single ovule. Now the question is wind pollination. What is the example given in NCERT? Wind pollination is shown by cereals. Wind pollination is shown by cereals. This is pretty important. Cereals. Cereals belongs to which family? Gramineae. Gramineae or Poaceae family. Wheat, maize, rice, right? These all are the members of Gramineae family. In them, the wind pollination will be seen, right? And that means wind pollination is seen by monocots. The members of the Gramineae family, all of them are monocots. Okay, this is important. So, wind pollination is shown by the monocots, the cereals, the members of the Gramineae family, and these are the characteristics. Now, let's go to the water pollination. Water pollination or hydrophily. Okay. Water pollination or hydrophily, that what is important regarding them, that pollen grains have, both pollen grains and stigma, both of them have mucilaginous covering around them, so that the water will not be able to cause any damage to the pollen grains and the stigma. So they have mucilaginous covering. Now, what is important regarding the water pollination is that this is very rare in angiosperms. Hydrophily is very rare in angiosperms. It is seen only in 30 genera. There are lakhs of species of plants present on Earth, and out of them, hydrophily is seen only in 30 genus of the angiospermic plants, only 30 genus. And amongst those 30 genus, mostly monocots are there. This is important line. Hydrophily is very rare, restricted only to 30 genera of angiospermic plants, and those plants are mostly monocots. Now, there is a question comes that in hydrophytes, hydrophytes are the plants which are present in the water. So, is this a common sense to say that hydrophytes will show hydrophily? No. Even in hydrophytes, hydrophily is least common, because most of the hydrophytes, they are present on the surface of water, and most of the hydrophytes, they show entomophily. Most of the hydrophytes, they are pollinated by insects, because most of them are present on the water surface. So, hydrophily is the examples of hydrophily are only very few, like Vallisneria, Hydrilla, and Zoosterra. These are the 3 hydrophytes or aquatic plants which show hydrophily. Otherwise, many hydrophytes, they show either wind pollination or insect pollination, right? So these are the 3 examples of hydrophytes which show a hydrophily. Okay, so many hydrophytes show either entomophily or anemophily, either wind pollination or water pollination. Sorry, and entomophily is more common. Insect pollination is more common in case of hydrophytes, like water hyacinth. You must have heard the name of water hyacinth, that is Eichhornia, and water lily. So in most of the hydrophytes, entomophily is seen, and in very few hydrophytes, hydrophily is seen. That is, I have given you the 3 examples which you have to remember. This is the diagram of NCERT. In NCERT, it is given that Vallisneria, this is the depiction of the plant Vallisneria, which show hydrophily. And in the Vallisneria, the hydrophily, the kind of hydrophily which is present is epihydrophily. What is the meaning of epihydrophily? That the hydrophily is occurring at the surface, just at the surface of water, right? These are the female flowers. These are the female flowers. And what happens? The female flowers, the stigma of the female flower is just present at the surface. It is not exposed to the air, but it is present just at the surface of the water. And the male flower will will be detached from the male plant, and they come towards the stigma at the surface, and the pollination will occur at the surface of water. This is epihydrophily. And Vallisneria again is a dioecious plant. That means male plant is different and the female plant is different. Okay. In Zoostera, hypohydrophily is seen. That means pollination occurs below the water surface. In Vallisneria, it is occurring at the water surface. It occurs below the water surface. Okay. And now comes the most important one. That is entomophily, or the insect pollination, What are the features? This is the most common type. What are the features? The flowers will have an odor, that is a smell. They will have color. They will have nectar. And the pollen grains of the insect pollinated flowers, they are very special because they have, remember, they have pollen kit around them. Who formed the pollen kit? The tapitum, which is a sticky layer, and they get sticked to the legs of the insect. Then stigma is also sticky. Okay, so these are the characteristics. If the flowers are small, generally the flowers are large sized. Generally, the insect pollinated flowers are large sized with bright color to attract the insect. But if the flowers are small, then they will be packed into inflorescence so that they will be easily visible to the insect. Otherwise, generally they are large sized flowers, but if they are small, they will be packed into inflorescence so that they can easily be conspicuous or visible to the insect. Okay, so these are the features of insect pollinated flower. Now, the insects, they do not have this feeling of doing the social service for the flowers. They do it for their own sake. That means they do it for the floral rewards. They get some rewards. They get some, something. Only because of that, they do the pollination. So what they get? They get nectar, the floral rewards. They get pollen grains, or they get the safe place to lay their eggs. The insects, they lay their egg into the flower, into the gynoecium of the flowers, so that their eggs, they get the safe place, right? Safe place to lay their eggs. Right? And the example of this, when the insect come towards flower so that they get the safe place to lay their eggs, is Amorphophallus. Amorphophallus, then fig, Portulaca. These are the plants in which the insect come to lay their egg. There is an association between fig and wasp. Fig and wasp. This is the obligate association. This is the obligate association. That means they cannot live without each other. Fig and wasp. Fig is the ficus, right? The plant, and wasp is the insect. They are so closely associated with each other that they cannot live without each other. And this is an obligate association. This question is also given this CBSE sample paper of term 2. Okay. So these are the plants which show obligate association. Now, so this is all about the pollination. Now, after pollination, what will happen? The pollination is done. Now, the stigma has, after pollination, what will happen? The pollen grain has landed on the stigma. This is the pollen grain. And it is present on the stigma of the carpel, right? So now what will happen here? Here at the stigma, there will be a pollen pistil interaction. There will be a pollen pistil interaction. Now, what is the meaning of pollen pistil interaction? The pollen grain will convince the stigma that I am the pollen grain of your own species. Please allow me to form the pollen tube. Then the stigma will say, okay, just a second, let me recognize you, whether you are of my own species or not. So the pollen grain carries the compatibility proteins formed by the tapitum and convinces the stigma. When the stigma gives the green light, the pollen tube will be formed. So pollen pistil interaction is the dialogue occurs between the pollen grain and the stigma. And when the compatibility is proved, when the compatibility is found, when the compatibility proteins are matched between stigma and the pollen grain, then pollen tube gets formed. Then pollen tube gets formed. And you know who will form the pollen tube? Who will form the pollen tube? The pollen tube will be formed by the tube nucleus, right? So this is the pollen grain. This was the germ pore, remember? Now the intine will be extended. Remember, pollen tube is nothing but the extension of intine. So here now pollen tube will be formed. And the nature of pollen tube is also pectocellulosic. Why? Because it is formed by intine. And who will help in the formation? The tube nucleus. This is the tube nucleus, the vegetative nucleus. It will help in the formation of pollen tube, right? Now the generative cell divides to give rise to two male gametes. Generative cell divides to give rise to two male gametes, right? So this generative cell will form two male gametes. So ultimately these are the two male gametes present in the, and these are non-motile. That means flagella is not present in them. The two male gametes, they do not have flagella. Okay. Now this is a mature male gametophyte when it forms the pollen tube and it carries the two male gametes. So who is the carrier of male gametes in case of angiosperms? The carrier of male gametes is the pollen tube. Who carries the male gametes? Pollen tube. And who carries the pollen grains? Many pollinating agents. So this is ovary. Inside ovary, what is present? Inside ovary, there is ovule, right? So this is ovule. And inside ovule, what we have just formed up? We have formed the embryo sac, which is waiting for the male gametes to come. So inside the ovule, So this pollen tube will grow towards the ovule, right? Towards the ovule. But who ensures that the pollen tube will enter into the embryo sac towards the micropylar side only? Who ensures this? Synergids. So now synergids will guide the pollen tube. Synergids will guide the pollen tube to enter into embryo sac, through micropylar side, These synergids, they guide the pollen tube that they have to enter into the embryo sac only through the micropylar side. Okay. And during this process, one of the synergids gets degenerated. One of the synergids gets degenerated before the entry of pollen tube. Gets degenerated before the entry of pollen tube. Just before the entry of pollen tube, out of the two synergids, one of the synergids gets degenerated. Okay. Now, what will happen? Ultimately, this is the embryo sac. The three antipodals, they will degenerate. The antipodals will degenerate. One of the synergids has already degenerated. One of the synergids has already degenerated before the entry of pollen tube. Right? This is the egg cell. And just before the entry of pollen tube, now the pollen tube is about to enter. The pollen tube is about to enter the embryo sac, and just before its entry, what will happen? That the two polar nuclei fuses. Polar nuclei gets fused to form secondary nucleus. Which is a diploid structure. So these two polar nuclei, which were haploid, they fuse to form the secondary nucleus. So this is the secondary nucleus. And then ultimately, the pollen tube takes entry into the degenerated synergid. The pollen tube takes entry into the degenerated synergid, and from the degenerated synergid, it takes entry into the cytoplasm of the other synergid, and ultimately the other synergid would also die. Okay, other synergid would also die. So what will be present here after this thing? That inside the embryo sac, nothing is left. Antipodals degenerated, both of the synergids degenerated. So now we have this secondary nucleus in the central cell. This is the secondary nucleus, and this whole is the central cell, right? Antipodals degenerated, synergids degenerated, and what is left here? Just egg cell. Antipodals also degenerated, and the pollen tube has released two male gametes. These are the two male gametes which are present in the embryo sac. So one of the male gametes will get fused with the secondary nucleus, and one of the male gametes will fused with the egg cell. So one of the male gametes So this is male gamete and this is secondary nucleus, and their fusion is called as the triple fusion. Their fusion is called as the triple fusion, right? Why triple fusion? Because two polar nuclei fuse to form secondary nucleus and then male gamete fuse to form the structure. That means ultimately three nuclei are involved, two polar nuclei and one male gamete, right? So triple fusion. Then other male gamete and egg cell. This is the male gamete, other male gamete, and this is the egg cell, and their fusion is called as syngamy. So within the same embryo sac, there are two fusions. One fusion between male gamete and secondary nucleus, other fusion between male gamete and egg cell. And this is called as syngamy, and triple fusion together is called as double fertilization. Within the single embryo sac, there are two fusions, that is called as double fertilization, and this is seen only in case of angiosperms. Okay. So what will be the result now? The result will be the fertilized egg cell. The fertilized egg cell forms zygote, and the fertilized central cell, the central cell got fertilized with the other male gamete, so the fertilized central cell forms PEC. What is PEC? PEC is primary endosperm cell, and this primary endosperm cell contains PEN, that is primary endosperm nucleus. And this primary endosperm nucleus is a triploid nucleus, 3N, because it was a result of triple fusion. Okay, so ultimately we have PEC, primary endosperm cell, and we have zygote. Now what we have to see is post-fertilization development, that is how embryo is formed from zygote and how endosperm is formed from PEC. So I hope up till now everything is clear to you. So now let's move on to the post-fertilization development. We will first see that how endosperm is formed from the PEC. So post-fertilization changes, and in the post-fertilization changes, let's discuss how PEC, the primary endosperm cell, which contains the primary endosperm nucleus, ultimately forms the endosperm, which is a triploid structure in the angiosperms. And what is the function of endosperm? To provide nourishment to the growing embryo. So, on the basis of kind of divisions occur in PEC, the endosperm can be of two types. It can be free nuclear endosperm. It can be free nuclear endosperm, or it can be cellular endosperm. Now, what is the difference between the two? In free nuclear endosperm, what happens that the pen initially undergoes many free nuclear divisions. And now you understand what is the meaning of free nuclear division, that only the nucleus will divide and not the cytoplasm. So after many free nuclear division, a multinucleated structure is formed. After many free nuclear division, a multinucleated structure is formed, and then ultimately in this multinucleated structure, the cell wall will form. Ultimately, the cell wall formation will occur and the endosperm will be formed. Right? That means initially there were only free nuclear divisions. Initially only nucleus divides, and when the cell contains many nucleus, then ultimately cell wall development occurs. This kind of endosperm is called as free nuclear endosperm, and this is most common type of endosperm found in angiosperms. Most common. Then what happens in cellular endosperm? The peck divides, and karyokinesis. After nuclear division, the cytoplasmic division occurs immediately. That means it's a kind of division where after nuclear division, the cytoplasmic division will occur immediately. That means karyokinesis will be immediately formed by cytokinesis. Here, karyokinesis immediately followed by cytokinesis. Okay, will be followed by cytokinesis. And this kind of endosperm is not very common in angiosperms. This kind of endosperm is seen only in some members of Solanaceae family like Petunia. Petunia is a member of the family Solanaceae, right? So these are the two major types of endosperms: free nuclear, cellular. In the free nuclear division, only in the free nuclear endosperm, most common, and cellular endosperm is seen in some members of Solanaceae. Now, there is a case called as coconut. Coconut is a case where two types of endosperm are present. In the same seed, two types of endosperm are present. That means the liquid endosperm, when you drink the water from the coconut, from the green coconut, the liquid endosperm, the liquid which you drink from coconut is nothing but the pre-nuclear endosperm. And the white kernel around it, the white portion around it, the white kernel around it, that is the cellular endosperm or the solid endosperm, right? So the white kernel, the solid part in the green coconut, which is white, that is the cellular endosperm, but the liquid you drink is the free nuclear. So coconut has two types of endosperm, right? Now let's see how embryo is formed from zygote, and that is called as embryogenesis. Formation of embryo from zygote. So in your syllabus, there is description of embryogenesis in case of diploid, right? But I'll tell you the case of monocots also. So let's take zygote. This is the zygote, right? Now, first of all, there will be unequal mitosis in the zygote. First of all, there will be unequal mitosis in the zygote, and what will be formed? Two cells will be formed. One is the larger cell called as the suspensor cell, and the another one is the smaller cell called as the embryonal cell. So it gives rise to two cells. Larger cell is the suspensor cell and the smaller cell is the embryonal cell. This suspensor cell will ultimately give rise to suspensor, and the embryonal cell will give rise to embryo. What is the function of suspensor? Suspensor will derive the nourishment from the endosperm, and the embryonal cell will form the embryo. So what will happen? This suspensor cell this suspensor cell, which is formed towards the micropilar side, the suspensor cell which is formed towards the micropylar side, this undergoes several divisions. This undergo many transverse divisions. What are transverse divisions? Like this. The periclinal or the horizontal divisions are the transverse divisions. So after many transverse divisions, the suspensor forms a 6 to 8 celled structure. And this is called as the suspensor. 6 to 8 celled structure. So in the suspensor cell, many parallel or periclinal or horizontal or transverse division occurs to form a 6 to 8 cell structure called as suspensor. And what is the function of suspensor? To derive nourishment from the endosperm and provide it to the embryo. This embryonal cell will undergo two longitudinal divisions and one transverse division. Two longitudinal, that means this is the embryonal cell. One division will occur like this, so two cells will be formed, and then another division will occur like this, longitudinal, 4 cells will be formed, and then transverse division. So 4 cells will be formed upper side and 4 cells will be formed towards lower side. So 8 celled structure is formed, right? So after 2 longitudinal divisions and 1 transverse division, how many celled structure is formed? 8 celled structure is formed. And this 8 celled structure, 4 cells towards the lower side and 4 cells towards the upper side. So this is an octent, right? Octent. And this octent is called as a pro-embryo. It is called as pro-embryo. Are you getting? Octent. So this is pro-embryo, right? Which has how many cell structure? 8 cell structure. So I hope until here the process is clear. Suspensor cell after many transverse divisions give rise to suspensor, and embryonal cell after 2 longitudinal and 1 transverse division give rise to the pro-embryo or the octent. Now, this octent, the suspensor will remain like this, but the octent will further, in the octent, many divisions will occur and it will form a globular embryo. After pro-embryo, globular embryo will be formed. Okay. Oh, before that, let me tell you some important thing. Before telling you the globular embryo, let me tell you about the octent stage. So 1, 2, 3, 4. 4 towards the upper side, 1, 2, 3, 4 towards the lower side. So this is an octent, right? So let's explain the various part of the embryo. This upper part up till here is the suspensor, right? And this is the pro-embryo. The uppermost cell of the suspensor is called as the hostorial cell. And what is the function of the hostorial cell? It derives the nourishment from endosperm. And the lowermost cell of the suspensor is called as hypophysis. Hypophysis is the lowermost cell of the suspensor. What is the function of hypophysis? hypophysis later on forms the root cap. It forms the root cap and a part of radicle. You know what is radicle. Radicle is present in the seed, which ultimately give rise to the root when you sow a seed into the soil. Then this is the embryonal octant. The 4 cells towards the hypophysis, these 4 cells are called as hypophyseal tire. Right? This is called as hypophyseal tire. And the 4 cells which are present away from the hypophysis, they form the epibasal tire. Hypobasal tire, sorry. This is called as the hypobasal tire. And this is the epibasal tire, right? The hypobasal tire will later on form hypocotyle, and it will form the remaining part of the radicle. It will form the remaining part of the radicle. And the 4 cells here, the epibasal tire, these 4 cells will ultimately form 3 main important structures. They will form first the plumule, and you know plumule give rise to shoot. Then it gives epicotyle and it gives cotyledons. Okay. So these are the various functions. The hypophysis of suspensor cell will give rise to root cap and the part of radicle. Hypobasal tire will give hypocotyle and remaining part of radicle, and these 4 cells, they will give rise to plumule, epicotyle and cotyledons. Right? Okay. Now, after further divisions in the embryonal octant, we will have the globular embryo. Now, globular embryo will be formed, and in this globular embryo, there will be different layers. The outermost layer is called as the dermatogen or the protoderm, and this dermatogen or protoderm will ultimately give rise to the epidermis. Then the next layer is the layer of the ground meristem, which ultimately will give rise to ground tissue. And as you have read the anatomy now, you know what is ground tissue. Everything except epidermis and xylem and phloem, everything is ground tissue. And inside the center, towards the center, procambium is present, which will give rise to the vascular cylinder, the xylem and the phloem. So these are the different layers which will give rise to the different parts, and now this embryo is called as the globular embryo. Now after further division, what will happen? The cotyledons will start to form. These are the cotyledons, right? They start to form. They start storing the result food material, and now this stage is called as the heart-shaped stage. And after further divisions, ultimately what will happen? We will have the cotyledonary embryo, and these are the 2 cotyledons. This is the mature embryo, which have proper 2 cotyledons. Right? These are the 2 cotyledons which you can see properly over here. And ultimately, the suspensor will degenerate ultimately. After providing nourishment to the embryo, the suspensor will degenerate ultimately. Right? And this embryo will give rise to a new plant. Okay? So, what question is asked? Arrange the various stages of embryo development. Arrange various stages of embryo development. And how will you arrange, beta? First stage is the proembryo. After proembryo comes the globular embryo. After globular embryo comes the heart-shaped embryo. And then ultimately the cotyledonary or the mature embryo. So these are the stages which generally comes to arrange. Okay. So these are the various stages in the development of embryo. Is it clear? Okay. Now everything in monocots also, everything happens as in dicots. In monocots, the embryo development is similar to that of dicots, but what is the difference? First difference is that in monocots, the suspensor is single-celled. Over there, the suspensor was 6 to 8 celled, but here the suspensor is single-celled. And the next difference is that there are two cotyledons here also. There are two cotyledons here also, but one is rudimentary. What is the meaning of rudimentary? Highly reduced. The second cotyledon is so much reduced that we do not even count it. That is why we say that in monocots only one cotyledon is present, right? So the second cotyledon is so rudimentary and reduced that it is not even counted, and that reduced cotyledon is called as epiblast. And the second cotyledon is developed. The second cotyledon or the single cotyledon, we count it as the single cotyledon because it is well developed. So the single cotyledon which is developed in case of monocots is called as scutellum. So if the question comes that the second rudimentary or highly reduced cotyledon monocots is called as, you will say epiblast, and the single cotyledon which is properly developed is called as scutellum. So apart from these differences, monocot embryogeny is similar to dicot embryogeny. Okay, now let's see the diagram of NCERT. Identify which one is the dicot embryo and which one is the monocot embryo. Obviously, this one is the dicot embryo. This one is the dicot embryo. You can see over here, this is plumule. These are two cotyledons. These are two cotyledons, right? This is hypocotyle, this is radicle, and this is root cap. This is a monocot embryo. And you can see that there is a single cotyledon. The single cotyledon is called as scutellum. This is the coleoptile, right? This is shoot apex. This is epiblast. See, the second cotyledon is so reduced that we do not even count it. Radicle, coleorhiza. Now, what are coleorhiza and coleoptile? Coleoptile is a special leaf-like appendage which surrounds the plumule. Plumule forms the shoot, and the plumule is protected by a leaf-like structure called as coleoptile, and coleorhiza surrounds the radicle. Okay, this is not present in case of dicot embryo. Coleorhiza and coleoptile is present only in case of monocot embryo. Right? Okay. So now let's see structures formed after fertilization. Structures formed after fertilization. What are the several structures that are formed after fertilization? So we have just read first endosperm. Now we know how endosperm gets formed up and what is the function of endosperm. Second, embryo, right? Then the third is the seed, then the fruit, right? So these are the structures which are formed after fertilization. So now let's discuss, first let's discuss the seed. A seed is called as a fertilized ovule. As soon as fertilization occurs inside the ovule no longer called as a seed or ovule, but it is called as a seed. So we can say that ultimately the integuments of ovule after fertilization, the integuments of ovule forms what? Seed coat. And as the integuments of the ovule form seed coat, and as there are two integuments, so that is why the seed coat are also two. The outer seed coat is called as testa and the inner seed coat is called as tegumen. Testa and tegumen. So an angiospermic seed has two seed coats, testa and tegumen. Right. And what is present inside the seed? The endosperm, the embryo. The embryo will give rise to a new plant. Okay. And what is another important point is hilum. Remember hilum? Hilum remain as a scar even on seed. A scar which can be seen on seed is hilum. And micropyle, the opening which was present in the ovule, it is also present in the seed also. Micropyle present, and through this micropyle the new seedling comes out, the new small plant comes out of this micropyle. Okay. Okay, so now this you must have read in the chapter morphology also, but let's see that types of seed. Seed can be of two types. They can be endospermic or albuminous seed. Oh, what's happening? Albuminous seed, or they can be non-endospermic or they can be non-endospermic. What is happening? Why this is overwriting? These technical things. Non-endospermic or non-albuminous. Non-albuminous or X-albuminous. Or X-albuminous. Okay. So what happens, beta, that when inside the seed the embryo grows, during the growth of embryo, if the endosperm is totally consumed up and it is not present in the seed later on, then the seed is called as non-endospermic seed. If the endosperm is totally consumed up by the embryo. If the endosperm is totally consumed by the embryo, then it is non-endospermic seed. And non-endospermic seed is mostly seen in dicots. That means in dicots, the embryo consumes the endosperm during development, and later on inside the seed endosperm is not present. But in monocots, what happens that mostly in monocots, the endosperm remains in the seed. That means the embryo do not consume up the whole endosperm during development. Right? So it remains in the seed, and it is seen mostly in monocots. So remember this, that mostly in dicots, what kind of seed is seen? Non-endospermic, and in monocots, endospermic seed is seen, right? But there are some exceptions to it, and these exceptions are important. What are the exceptions? First, orchid. Orchid is a monocot, right? So what sort of seed should be present in it? Endospermic. But no, it is a monocot, but still it has non-endospermic seed. So these exceptions are important. Then second is castor, coffee, or sunflower, right? These are dicots. These are dicots, but still they have endospermic seed. So in dicots, non-endospermic seed is found, but these are dicots, but still they have endospermic seed. And orchid is monocot, but still it has non-endospermic seed. Okay, this is important. Then another kind of seed which is important is perispermic seed. Then what is this perispermic seed? The seed in which perisperm is present. The seed in which perisperm is present, that is called as perispermic seed. Now what is perisperm? Perisperm is remains of nucellus. When there is fertilization in the ovule, during the development of ovule, the nucellus gets consumed up. Nucellus is never found in the seed. It is all consumed up. But sometimes nucellus remains in the seed, and that nucellus which is present in the seed is given an important term that is called as perisperm. So perisperm is nothing but the remains of nucellus which are present in the seed, right? And perispermic seed are found in plants like castor. Castor has a perispermic seed. Then black pepper. Then beet. Okay, so these are some examples of perispermic seed. Okay, so this makes an important question. Now, seed show two properties which are very important. Seed show two properties which are very important. First is dormancy, and the second one is dehydration. A seed will not germinate until and unless it gets all the requirements fulfilled, both external and internal. Until then, the seed will remain dormant. And when the seed matures, it loses its water, right? It gets dehydrated. Both of these qualities are very good for a farmer because of these two qualities, a farmer can store the seed, right? Because it will not germinate until and unless it will get the favorable conditions. So it is easy to store it, and it gets dehydrated because wherever there water is present, water gives gives the possibility of pathogenic infections. But because the seed show dehydration and dormancy, these are very two important properties for farmers, right, to store seed. Because of these two properties, the farmers can easily store the seed. Okay, so these are the properties of. Now let's move on to the another topic, that is seed viability. Seed viability is the capacity of the seed after the seed is formed, then for how much time after the formation of seed, for how much time the seed is capable of giving rise to a new plant. That is called as the seed viability. And some plants lose the capacity. Some plants have seed viability only for few, months, like oxalis. The seed is formed and within few months it will be able to give rise to a new plant. After that it will not be able to give rise to a new plant, but it can be up to several years. One example is of lupine. It was excavated from Arctic tundra, right? And when it was excavated from Arctic tundra, with the help of carbon dating system, its age was calculated and it was 10,000 years old. And even after excavation, when it was sown, it gave rise to a new plant. That means this plant, lupine, has a seed viability of 10,000 years. And the second example given in NCERT is of the date palm. Also called as, the scientific name is called as phoenix. It was excavated from Dead Sea, and the seed which was excavated was 2,000 years old, and the seed, even after being so much old, gave rise to a new plant. That means the seed viability can be up to several years. Okay. Then what is formed after fertilization? Fruit. And what is a fruit? A fruit is a fertilized, ripened ovary. Fertilized, ripened ovary. After fertilization, the ovary gets ripened up to form the fruit. Right. And the ovary wall, the wall of the ovary after fertilization give rise to the fruit wall. And the fruit wall is called as the pericarp. The pericarp, right? Now, the fruit can be of three types. This you must have read in the morphology. The fruit can be a true fruit, it can be a false fruit, or it can be a parthenocarpic fruit. True fruit is a fruit in whose formation only ovary is involved. The fruit in whose formation only ovary is involved, that is a true fruit. But false fruit is a fruit in which along with ovary, other floral parts are also involved, like thalamus or inflorescence. So along with ovary, other floral parts are also involved, like the apple, the pear, the strawberry, and most of the fruits are the true fruits. And the parthenocarpic fruits are the fruits which are formed without fertilization. Right? This question is coming NEET. Fruits which are formed without fertilization, and the example is banana. Okay, so these are all the structures which are formed after fertilization. Now the last topic of the chapter, and that is apomixis. Before apomixis, let me show you some diagrams of NCERT. Yes, this is the diagram of the seed, right? This is the seed of monocot. What you can see here in the seed of monocot, seed of monocot, this is endosperm, and around the endosperm there is a special layer. Around endosperm there is a special layer called as the aileron layer, right? which has some important enzymes, hydrolytic enzymes, which helps in seed germination. Scutellum, this is the single cotyledon, right? Coleorhiza, coleoptile, and this is the seat of dicot. Okay, so these are the diagrams of NCERT. Now, let's discuss what is apomixis. Amphimixis means the division of gametes. Amphi means not division, fusion. Amphi means two, mixis means mixing. When the two gametes gets mixed up, they form zygote, right? And the zygote forms the embryo. This is normal sexual reproduction. But what is apomixis? Apomixis is formation of seed without fertilization. Without fertilization. When do you call a ovule a seed? As soon as within the ovule an embryo is present, then that ovule is not called as ovule but seed. If that embryo is the product of fertilization, then the seed is the proper sexual seed. But sometimes what happens that inside the ovule, embryo forms directly from integument or from nucellus. Directly. Embryo forms directly from integument. Integument, remember the coating of the ovule, or nucellus, the parent gametous tissue present in it, right? Without fertilization. So that ovule will now be called as seed, but the seed is not the product of sexual reproduction, but it is formed asexually. So what is called, apomixis is called as formation of seed without fertilization, right? And this is a type of asexual method. This is line of NCERT. This is the type of asexual method which mimics sexual reproduction, because formation of seed is a sexual reproduction, right? So apomixis is asexual reproduction which is trying to be sexual reproduction. Okay, this line is important. In NCERT, there are two families which are given in which apomixis is commonly seen. Two families in which apomixis is seen. And these two families are Asteraceae family and the Gramineae family. Gramineae or Poaceae family. Asteraceae family is the sunflower family. Sunflower, marigold, chrysanthemum, zinnia, right? And this is the family of cereals. So apomixis, this is important. Apomixis is formation of seed without fertilization. It's a type of asexual method which mimics sexual reproduction, and it is commonly seen in two families, Asteraceae, the sunflower family, and the Gramineae, the cereal family. Okay. Now what happens here? The embryo is formed without fertilization inside the ovule. And this gives rise to polyembryony. What happens in the seeds of some plants, that inside the embryo sac, the normal embryo, which is a product of sexual reproduction, forms. But an extra embryo also gets formed up by nucellus or integument, so that the seed contains more than one embryo. So polyembryony is a phenomenon where seed contains more than one embryo. Seed contains more than one embryo, and the extra embryo, the main embryo which is formed inside the embryo sac, is the product of sexual reproduction, but the extra embryos are the product of apomixis, right? How? See, the extra embryos which are present in the seed are formed directly through nucellus or integument without fertilization. This is apomixis, right? So due to apomixis, polyembryony is seen in some plants where the embryo is formed without fertilization. The extra embryos are formed without fertilization by directly from the cell of nucellus or directly from the cell of integument. And this kind of polyembryony is called as adventive polyembryony. When the extra embryos are formed either by the nucellar cell or the cells of the integument, that is called as adventive polyembryony. And this adventive polyembryony was first discovered by Anton von Leeuwenhoek. Polyembryony was first discovered by Anton von Leeuwenhoek in citrus fruits. Citrus fruits are lemon or orange. So polyembryony was first discovered in citrus fruits and it is also found in mango. So this question often comes that in citrus fruits and in mango, the extra embryos comes from which part? So you will go say nucellus or integument because they show adventive polyembryony. Clear? So this is polyembryony. Now there are some advantages of apomixis given in NCERT. And what are those advantages? Because if you find the scientists are searching on the apomictic genes, the scientists are working on the apomictic genes, if they will get the genes which are responsible for apomixis, then that would be a revolution in the history of the plant breeding program. Because if the scientist will find the apomictic genes, they will insert those apomictic genes in the hybrid varieties, in the hybrid seeds, right? And when they will inject the apomictic genes into the hybrid seed, then that seed will form the embryos only by asexual reproduction and not by sexual reproduction. Then that hybrid plant will give all the seeds by asexual reproduction and all the seeds will be similar to the parent. So the farmer will not have to buy the hybrid seeds again next year because there will be no segregation of traits. Why? Because the apomictic genes will not allow the segregation of traits. Segregation of traits is seen during sexual reproduction, right? But when due to apomixis the embryo will be formed, there will be no segregation of traits and the seed will be clone of the hybrid plant. So that would be very easy for the farmers. They will not have to buy the They will not have to buy the seeds every year, right? So what is seen here, what we can see here in the NCERT, what question comes from this point? Let's see. Let's see here. It's saying that hybrid varieties of our food crops are being extensively cultivated. Cultivation of hybrids has tremendously increased. We know this, that hybrids are, hybrid seeds are sown so extensively in agriculture these days. One of the problems of hybrids is that hybrid seeds have to be produced every year. That means farmer have to buy the hybrid seeds every year from the market. Why? Because the hybrid seed he has taken from the market, he will sow it in the field, then it will form a hybrid plant. Then if that hybrid plant will sexually reproduce, then all the seeds formed on that plant will not be similar to the hybrid plant, right? So the farmer do not take the risk of sowing these seed in the next year, because these seeds will be the product of sexual reproduction and they will not be similar as the hybrid plant. So he will buy the seed again in the next year. But if we will inject the apomictic genes into this hybrid variety, then all the seeds will be formed apomictically. They will be cloned to the hybrid plant and the farmer will not have to buy the seeds again and again every year. So there's a hot research in the apomictic genes. If they will be found, they will be injected in the hybrid seeds. If the seeds collected from hybrids are sown in the plant, they will segregate and do not maintain hybrid characters, right? Generally, what happens, the seeds, they do not retain the characters because they are product of sexual reproduction, because there is segregation of traits. But if these hybrids are made into apomicts, but if we inject apomictic gene into these hybrids, there is no segregation of characters because there will be no sexual reproduction, and that is why farmer will not have to buy the seeds every year. Okay, so this will be the advantage of discovery of apomictic genes. So I hope you have understood the chapter well. All the points of NCERT are covered. Listen through the lecture carefully, then read your NCERTs and then solve the questions, and your chapter will be prepared. So we'll meet up in the next chapter. Until then, bye-bye. Love you all.